And thanks for your patience, everybody. We had to swap a computer here to get things to work right. And Dale is,
Did it faster than I would.
so why don't you step up there and I'm going to ask you 5 questions.
Okay.
Here we go. Yeah. Dale, where were you born?
Milwaukee, Wisconsin.
And where'd you go to high school?
John Marshall, Milwaukee.
And then where'd you go for your undergrad and what did you study?
University of Wisconsin, Milwaukee. And I studied chemistry.
And then where'd you go for your advanced degrees and what'd you study?
From there, I went to Indiana University where I earned my PhD. And then a postdoc at the Argonne National Lab outside of Chicago.
And you've always done chemistry, not biochemistry.
Right.
Because you do
Yeah.
close to biochemistry to me, but you're
Well,
trained
I
in
cross over a little bit in clinical chemistry.
yeah. And then when did you come to UW-Madison?
1997.
And you were in nutritional sciences?
Yes.
One of the 33 programs and departments started by Stephen Babcock.
Mhm.
All right, maybe it's just 5, but yes.
Yeah, well,
And then
I'll go from there.
and then you retired or
Semi-retired, yeah.
and semi-employed?
Professor emeritus at the university in nutritional sciences, but I'm still running a core lab in the Biotech Center.
That's amazing. Thank you for doing that. And you're on the what floor at the Biotech Center?
Second floor.
So if you want to come and see the cool stuff, this is it. Today you have this great thing about sleep and weight management. Would you please join me in welcoming Dale Schoeller to PLATO.
Thank you, Tom. So I'm going to talk a little bit about the bioenergetics of weight control. For studies that we do in the stable isotope lab. Oops. That worked. All right, so we work with the light elements, hydrogen, carbon, nitrogen, and oxygen. And let's take carbon as an example. So carbon is made up of 2 stable isotopes plus 1 radioisotope, carbon-12, which is most abundant isotope, making up about 99% of all the carbon in the world. And carbon-13, a little over 1% of the pool of carbon on Earth here. And carbon-14 is a very minor isotope, radioactive. But we're interested in measuring the ratio of carbon-13 to carbon-12 in a lot of biochemical compounds and use isotope ratio mass spectrometry. Isotope ratio mass spectrometry is interesting because it has a huge advantage in terms of precision. So we'll show a sample here, which might come into the lab, which has a ratio, lost a decimal point there, of carbon-13 to carbon-12 of about 0.011. But with the isotope ratio mass spectrometer, we can measure down to the 5th decimal place here. So we can look at differences between 0.011231 and 0.11230. It's, where am I? Okay. Oh, there. Thank you. My reading glasses are not quite strong enough to see those small ones. So, but in terms of talking about ratios that are this similar, it's rather difficult. So we use a unit called the per mil unit, parts per thousand unit. It's sort of a percent difference between 2 carbon samples times 10. But the delta or del per mil of whatever we're measuring is expressed as the ratio of the sample that, of the sample that we're measuring there's pointer, divided by the carbon-13 to carbon-12 ratio of an international standard or a reference carbon, minus 1 times 1,000. So it's kind of like calculating percent difference times 10. Show two examples here, two samples relative to an international standard, which is limestone sample from PD County in South Carolina, or, in the Atlantic area, Atlantic Ocean area of the US. But differences between samples 1 and 2, which are shown up here, converted to a per mil unit. So sample 1 is 0.64 per mil lighter than our international standard, and sample 2, 2.42 per mil lighter than our international standard. But those are easily measured, and the difference between them by isotope ratio mass spectrometry, because we can measure to the 0.01 level. Now, an isotope ratio mass spectrometer, in terms of mass spectrometry, is not very exciting. It's low mass. It kind of handles masses between 1 and 70. Not much for biochemical compounds, which proteins, now you're talking thousands of mass units. But it's ideally set up for the light elements in their gaseous form: hydrogen gas, carbon dioxide for carbon and oxygen, and nitrogen gas. And as I just tried to point out, they're very high precision for measuring very small differences in the ratio of carbon-13 to carbon-12, or oxygen-18 to oxygen-16, or deuterium to hydrogen or protium. It's a mass spectrometer. These, oops, jumps too easily. The gases are introduced through an inlet, say a gas bench, so we can sample a gas sample in a glass tube, stoppered with a septum seal, so we can punch through that. We can introduce the gases from an elemental analyzer, which take a dry bulk organic material and burn it to CO2, nitrogen gas, or a chromium reduction system, which converts water to hydrogen gas. And they're introduced to the ion source. The ions are formed, accelerated out through a magnetic field. They follow a curved path. And then the masses of interest are projected out onto a Faraday cup collector, which is nothing more than a non-amplifying collector that sits there, and the ions impinge on it. Electrons flow back through there, neutralize the ion, so that you get a current which is equal to the flux of ions coming through the mass spectrometer. And that's fed into a computer and converted to the isotope ratio. What's interesting about these stable isotope ratios is that there are some differences in nature which can be used as biomarkers of various life processes. For example, nutritional epidemiology field that does a lot of work trying to determine the relationship between a person's diet and the odds of developing heart disease, cancer, stroke, diabetes, hypertension, Alzheimer's, based upon collecting their reports on what they eat. So they're given a diary, either on a computer now or on paper. They write down what they had for breakfast, snacks, lunch, dinner, snacks throughout the day. And that's converted into their food intake and their nutrient intake. However, people aren't very good at actually reporting their actual dietary intake. This is a plot of dietary data from healthy weight individuals, what they report eating in terms of the calories per day, and what is measured using a objective, accurate method to measure energy expenditure, assuming that they're not gaining or losing weight. Their intake should equal their expenditure. People tend to forget many items when they're reporting their dietary intake. How about that? I know I can't do it. But even healthy weight individuals underreport by about 10–15%. Oh, that's weight loss. Those are obese individuals who are trying to lose weight. They say they do better in terms of following the diet than they actually do. The normal controls underreport a little bit. Obese controls underreport about 20% of their caloric intake. And if you look at what foods they're reporting, there's a way to accurately determine what their actual protein intake is by measuring the nitrogen output in urine. So while an obese individual underreports by about 25–30% here, in terms of total calories, they're only underreporting about 10% in terms of protein, which means they're dramatically underreporting either their carbohydrate intake or their fat intake. Don't know which yet. In terms of carbohydrate intake, though, we can use the carbon isotope ratio mass spectrometer to look at some foods. There are two types, or actually three types of plants in nature. C3 plants that fix carbon using the Calvin cycle, and C4 plants which fix carbon using the Hatch-Slack process. But what's interesting is the two types of plants differ in terms of their carbon isotope ratio. Oops, it's too jumpy. So that a number of C4 plants in our food chain, interestingly, corn, sugarcane, sorghum are C4 plants. They're the primary constituents of sugar in our diet. So they're isotopically heavier than all the other carbohydrates that we take in: wheat, rye, various other carbohydrate sources.
Do any plant people know how the plant discriminates between 13 and 12?
That's been worked out. Can I give it to you right now? No, but the largest discrimination that's seen in the C3 plants comes largely from the velocities of CO2, heavy and carbon-13 containing carbon dioxide in the air, moves slower at the same energy level in the air, moves slower and hits the surface of the plant, the uptake mechanism, less frequently compared to its population in the air. The carbon-4 plant has been worked out biochemically, but I can't remember right now what the difference is in fixation.
So I apologize if misspoke. I said the C4s discriminated, and it's actually
They-
the C3s that prefer.
Well, they both the C4 discriminates less. The atmospheric CO2 is about -8 per mil. So they do a pretty good job of reflecting the CO2 that's in the air, where the C3 plants are quite a bit different. And that's been used by Dr. O'Brien in Alaska. She looked at Native Alaskan children. Rather than asking them how much sugar they were eating, she actually measured it in terms of the isotopic ratio of carbon that's fixed in alanine. And on the x-axis here She has the calculated added sugar intake versus the number of dental caries in terms of portion of tooth surfaces that are decayed in their mouth, and was able to show that indeed the more sugar intake that they were consuming in terms of its as measured isotopically as a biomarker, the more caries, the more surface areas that were decayed. But I don't want to talk about that so much. What I want to talk about in getting into our sleep study was the measurement of total energy expenditure. As you all know, in the last 100, 150 years, the average weight of citizens of the United States has gone up and never seems to come back down. But based upon Army records here, starting at the Civil War and going through the Korean War, the average weight of a 5'10" recruit has gone up from about 147 pounds in the Civil War to about 165 pounds during the Korean War. And then the US began their national surveys of health on health. And during that recorded height and weight of a subpopulation of US citizens that were selected randomly. And in the last 50 years or so, it's been a dramatic increase in the rate of the average weight from about 165 to 194 pounds. One of the common hypotheses for obesity that was put forth in the 1950s and is still talked about today as the cause of obesity is that individuals with low energy intakes, because they have a low energy, that report low energy intakes, must have a low energy requirement. And that's just overpowered in today's world where you can't go much any place in an American city and be more than 50 to 200 steps away from some source of food. And if you ask people how much they eat, I did say self-reported intakes tend to underreport their intake. But based upon that data, the epidemiologists and others suggested that, it must be a low energy requirement that's causing these people in this modern day to overeat because there's just so much food opportunities to eat. However, when we applied this measurement technique, which I'll talk about shortly, this doubly labeled water technique for measuring a person's energy expenditure, paper was published in 1986 by a group in England, headed by Andrew Prentice. Individuals with a BMI of greater than 30 kilograms per meter squared, which is the definition of obesity based upon height and weight, compared to a healthy weight individual or a group of individuals with a BMI of less than 25 kilograms per meter squared. Those that were of higher relative weight, higher BMI, were actually expending about 500 calories a day more than the healthy controls, which is not consistent with that hypothesis of low energy requirement, and certainly not consistent with the epidemiologic data on dietary intake for calories. The method, this doubly labeled water method, began with a study done in 1949 by Nate Lifson. He was a physiologist, was interested in finding out where the oxygens in expired CO2 came from. Did they come from the air that we were absorbing through our lungs, or the oxygen we were absorbing through our lungs, or did it come from body water? So he did 2 studies in a group of lab mice One in which we provided them with oxygen 18 labeled oxygen, molecular oxygen. And a second study in which he injected some oxygen 18 labeled water into the mice and labeled their body water. And what he found is that the oxygens in expired CO2 were actually in equilibrium and exchanging with the body water. So they were being labeled by the water. That gave him an idea. He hypothesized that the kinetics of hydrogen and oxygen in the human body or in any living animal could be used to measure the rate of CO2 production, where the pool size times the rate of elimination of a labeled hydrogen from body water would be equal to the rate of water turnover in that individual. And the pool size, N, times the oxygen elimination rate would be equal to the rate of water turnover plus 2 times the rate of CO2 production. 2 is in there because there are 2 oxygens in each CO2 compared to 1 in water. 2 equations, 2 unknowns. You can solve for the rate of CO2 production and come up with the rate of CO2 production is equal to the pool size, the total body water pool size, divided by 2, times the difference in the elimination rates. It seemed entirely too simple, but he was able to test his hypothesis in 1955. I don't have that slide. He did it in a group of lab mice, 10 lab mice, and showed that indeed this kinetic analysis using doubly labeled water could be used to measure CO2 production in the mice compared to what was actually measured when they were put in a cage and the CO2 was collected from their exhaled breath. In the human, it's the same thing. You have the individual drink this doubly labeled water. It's about $500 worth of isotopes. So we can't quite do it on everyone yet. That mixes with the body water pool over about 2 to 3 hours after they drink it. It's very quickly mixed body water pool. And then you collect sample of body water, either blood or urine is the easiest to take. The day of the dose administration, and then you can do it daily, or in this case, they did it at the first, second day after, and third day after the dose. And then about 2 weeks later, you're down about 2 half-lives, so you're down to about 25% of the initial enrichment of the double labeled water that after they drank it. And you can calculate these elimination rates. And you get the total body water pool from simple dilution. You know how much isotope you gave. You know what the concentration, the enrichment is in the body water pool after they drank it. You can calculate the pool sizes, which means you can be, in theory, measure the CO2 production of these individuals over about a 1 to 2 week period. You basically turn the body of the animal into a metabolic recorder. Fortunately, it had never been used, the method had never been used except in small lab animals, because when Nate Lifson was measuring the oxygen 18 enrichments over time after he dosed them, he had to use an amount of isotope that, if scaled up to the human, would cost about $50,000. And NIH is a pretty good source of funding, but it's not enough to buy everyone a new car. So I read his papers. I had an isotope ratio mass spectrometer. I said, let's try it. We tried it in 4 subjects, and we found indeed it was an accurate method for measuring energy expenditure with an accuracy of about 2% and a precision of about 6%. The nice thing is If you measure CO2 production, you can calculate how many calories they used over that period of time. Since then, there have been a number of labs which started to use this method in humans. There are about 6 validation studies in which subjects were their energy or CO2 production was measured by double water over a 5 or 6 day period while they lived in a bathroom-sized indirect calorimeter, which they actually collected their expired CO2. The accuracy with combining all of those 6 publications, gave you about 61 healthy adults, overweight, body mass, about 80 kilos. The method was accurate to better than 1%, precision about 8%. The equation that's used is a little more complex than the one that Nate Lifson was using because we had to add in factors for isotope fractionation as it exchanges into the CO2 and as it evaporates from the body water. And then that CO2 production is multiplied by 22.26 to convert. To convert the moles of CO2 production to the liters of CO2 production. And from that, you calculate the calories. In addition to measuring energy expenditure, one can use the double labeled water technique also to measure energy intake. That's been a great unknown in human research, very difficult to do unless they're actually living in your research facility and you're feeding them everything they eat. But once they go home, you have to depend upon them telling you what they eat, and we know they aren't very good at that. So we can measure energy intake using the caloric or the energy balance equation. Energy intake is equal to energy expended plus the change in your body energy stores. How much fat or fat-free mass you gain or lose during this 2-week period. The equation is shown down here. Fairly straightforward. The method of measuring energy intake has actually been validated in about 88 subjects, shown to be about 2% accurate and have a precision of about 7%. But the precision is very dependent on how well you measure the change in energy stores. There's no perfect method unless you actually do a chemical analysis of the individual. But to measure change, you need to do that twice, and you can only do it once in the volunteer, assuming they're willing to donate their body to science. But using here bioelectrical impedance analysis, there, it's a very imprecise method. You can't measure change in body fat very precisely over a 2-week period. So it's not very good at trying to measure energy intake over a 2-week period. DEXA, dual energy X-ray absorptiometry, a lot better. And now a new technique, quantitative whole body magnetic resonance imaging. You can get the precisions down to the 7% range. But the validations have shown it's up to 37% with the poor techniques for measuring body composition. Okay, so we have, with using isotope ratio mass spectrometry, validated techniques for measuring energy expenditure, and if we combine that with body composition change, validated techniques for measuring energy intake. So we can really start studying obesity, its causes and treatments with good, objective, accurate techniques. Okay. The CDC, though, has, of course, trying to stop the obesity epidemic as best it can. And it has major suggestions for achieving and maintaining calorie energy balance, preventing weight gain over time. And it suggests healthy eating, which I'm sure some of your relatives have suggested to everyone. Being physically active, also a common suggestion. And optimal sleep. And I've read these for 20, 30 years. And healthy eating and physical activity makes sense in terms of energy balance and weight gain. I mean, if the calories that you consume are equal to the calories that you expend, you got to maintain a constant weight. If one exceeds the other, you'll either gain weight or lose weight. But where does sleep come in here? So I ignored that recommendation for about 30 years. Yet there was, at that time, some reasonable data which suggests there's some relationship between sleep and what your BMI is, what your weight for height is. This was questionnaire-based data, and they divided the individuals up into those that slept less than 5 hours a night, 5 and 6 hours a night, 6 to 7, 7 to 8, 8 to 9 hours a night, and greater than 9. And what they found is about 8 hours to 9 hours of sleep a night was associated with the lowest BMI. So there must be something about sleep which is influencing.
They switch between cause and effect.
It's an association. So it's not, it's a good question. It could be either way. But you start to look at it in terms of doing some controlled studies. And there have been a number of controlled studies over the last 10 years. It's getting up to 20 years now. Someone wants to come in. What? I give it an okay. And what they did is, under laboratory conditions, they brought people in and they woke them up after about 4 hours of sleep so that they were sleep deprived, sleeping far less than that 8 hours per night. And then either at one meal thereafter or a day or two following, they were fed their food cafeteria style, large family-sized bowls, and they determined how many calories they ate, both during their normal sleep pattern of 8 hours and their sleep deprivation state. And they found that the people, after being deprived of sleep for 1 to 3 days, would overeat by about 300 calories a day. And in non-randomized trials, which are not quite as strong an evidence, they found about 400 calorie per day difference between those who were sleep deprived and when they were allowed normal sleep. During those studies, they looked at a number of hormone systems, and they found that indeed sleep deprivation caused an upregulation of ghrelin, which is a hunger hormone, causes increased energy intake, and a decrease in leptin, which actually causes, the decrease in leptin also causes hunger or reduces satiety, so people tend to overeat. But there are also mechanisms involved in the reward system, so that sleep deprived individuals are looking for a reward, a gold star, something like when your grandchild gets a gold star for doing good homework. That makes you feel good. In our case, piece of chocolate cake. gives us a little reward. Certainly works for me. So there are the biochemical mechanisms of that overeating after sleep deprivation are beginning to be worked out. Let's look at the epidemiologic data on sleep deprivation in the US. In the 1960s, the average American, according to the American Cancer Society, slept 8 to 8.5 hours a night. Similar studies done in 1997 found that sleep duration in the US averaged not 8, but now 7 hours a night. 2005, 30% of adults reported, less than 64 years of age, reported sleeping less than 6 hours a night. 1990, average sleep duration on a study done there was 6.2 hours a night. 2003, another one, the cardiac study of cardiac health, found that sleep duration was 6.1 hours per night on average. So this dramatic decrease has occurred across this same period in which weight has gone up. And if indeed these laboratory studies are predictive of what happens during their normal living conditions at home, it could be accounting for a major part of the weight gain over the last 50 years in the US. Seven years ago, Ezra Tassali, an MD at the University of Chicago, where I had worked for the 20 years before I moved here to the University of Wisconsin, proposed a study. Well, can we reverse this issue of overeating by bringing people in that habitually sleep less than 6.5 hours a night and counsel them to sleep 8 hours a night, and then send them home and measure their energy intake during a control period in which they're sleeping their short sleep of 6.5 hours a night, and their now longer sleep, whatever it is. So I want to test that. We put together a study design in which we recruited young adults, 21 to 40 years of age, BMI in the overweight range, so a BMI of 25 to 29.9, and who reported that they slept less than 6.5 hours a night. And that 6.5 hours of sleep was confirmed by having them wear a wrist activity monitor. And it turns out when you fall asleep, you move your wrists less. And actually, from wrist movement, you can detect when they fall asleep and wake up and measure their sleep duration. Excluded anyone in that study who had obstructive sleep apnea, which causes waking during the night, insomnia, or a history of other sleep disorders, or who were engaged in or had been recently engaged in shift work, in which they altered their sleep patterns depending on the day of the week. Recruited 80 individuals, which were randomly divided between a control group in which there was no change in no instruction to change their sleep time, and a group which would then be instructed to sleep longer. The groups were well matched in terms of age, sex. BMIs were very similar, identical between the two groups. Race and ethnicity, they were about half Caucasian, half other races or ethnicities, and excluded individuals who exercised at a high level, but did not exclude those who, only did long walks or maybe go to the gym for half an hour, a couple times a week. So about half the subjects in each group were moderately active, a fairly representative group. And the sleep averages during that week of confirming their sleep was about 6 hours a night in both groups. The design is shown here. The subjects were first studied for 2 weeks following their habitual sleep pattern, and then they were randomly divided. In the group that was in habitual sleep was not given any counseling on sleep hygiene or sleep elongation and continued to sleep their normal patterns. The other group spent about an hour, hour and a half with Dr. Tasali, who helped clean up their sleep hygiene, instructed them to have a quiet period of about half an hour before their programmed sleep time, and then removed all the electronics from their bedroom. No phone, no computer, which was the major cause of their short sleep. Took that out of their bedroom, and then both these groups were studied at home under the normal living conditions, not in the laboratory. They were told nothing about measuring their caloric intake using this combination of doubly labeled water and DEXA for body composition change. Then we also did some indirect calorimetry where we had them breathe into a respiratory gas analyzer and measure their CO2 production and oxygen consumption in terms of their resting metabolic rate, thermic effect of a meal, and they were given several questionnaires. As I said, the individual sleeping session was with Dr. Tasali, and most cases, she pointed to their electronic use at bedtime, or actually beyond bedtime, which would keep them up late. And shown here are their sleep durations as measured by the wrist activity monitor. Oops.
Boy, it went up that fast from 9:13 to 9:15.
Right. One session, one hour session of cleaning up, of quote unquote sleep hygiene, cleaning up your sleep conditions, had a huge effect. And they sustained it for 2 weeks. And the interesting thing shown here, the effects of the sleep intervention, comparing the sleep elongation of the control group, using our objective measurement, not their self-reported dietary intake. They were given no instructions on losing weight, eating less. That was kept hidden from them. And actually, I had a little problem with the IRB, said, gee, you can't measure something and not tell them that you're measuring it, but we did. They'd let us.
How did you justify that?
Just that knowing what they ate, would have no health consequences on those subjects. Yeah, so they allowed it. Energy intake of the sleep elongation group without any prescription to eat less decreased 270 calories a day. Yep, or 5 Oreo cookies in my case. Total energy expenditure went down a little bit, but not much. It was not significant. Resting metabolic rate changed by a significant amount, but only 33 calories, but it was statistically significant. Thermic effect of meals unaffected. Activity energy expenditure, no change. So the main effect of the intervention the sleep elongation was to eat fewer calories per day. And if you take that 270 calories, and if that is maintained for, say, a year, which is untested, it's an assumption, that would lead to about a 12 kilo weight loss, about 25 pounds. Probably not going to happen, but if you assume that nothing changes, that's what would occur. Would be a major treatment effect of sleep elongation. This is the intake data. It's a little noisy because of measuring body composition in a 2-week interval is not precise on an individual level. But as a group, you can see that in the control group, their intake actually went up a little bit on average. Oops. And in the intervention group, it went down. The increase in the control group is not unusual. You bring people into any study, even if you don't tell them, you're looking at their energy intake. They know that they're being weighed daily. They were given a Wi-Fi connected bathroom scale, so they knew we were monitoring their weight. And just telling people that you're monitoring their weight, they start to clean up their act a little bit and start to eat a little healthy. That usually goes away after the first couple of weeks. So the other group decreased their intake without any prescription to do so. And what was interesting is there was this reasonable correlation between the sleep duration elongation, well, sleep duration during the second 2-week interval and the energy intake that we measured in both groups using this combination of body composition and total energy expenditure was fairly strong. And the average increase in sleep was 1.2 hours. Or, yeah. Which, as I say, could translate to a significant weight loss if maintained over a 1-year period. And one of the things that they were asked in the questionnaires were, are you willing to maintain this lifestyle change to sleep over 7 hours a night? And most of the subjects, about 2/3 of the subjects, 3/4 of the subjects said, yeah, I'm about 60% confident that I could do this for a long period of time.
Is that subject number?
Yes, that,
Okay, so that's a name.
right. Yeah. I first just numbered them from 1 to 80, and then when I unscrambled them and lined them up this way. But that's just a way of tracking them without releasing their names to the audience. So the conclusions of this study were rather dramatic, and it was published in the Journal of the American Medical Association and picked up by many dozens of newspapers and radio stations and TV stations around the US. They now track that for you. That a modest sleep hygiene training, hour and a half, with an expert in the field, Dr. Tassali, increased their sleep to 7.1 hours per night from 6.5. Although we only measured that for a 2-week period. They think they could hold it longer. We don't know. And when we measured energy intake using an objective and unannounced measure of energy intake, it decreased their energy intake by a significant amount. Snickers bar a day. Actually, Snickers bar is about 180 calories, so it's 1.5 Snickers bar. Well, this is true, yeah. The king size is up there. It was interesting that the change in energy intake was very similar, but in the opposite direction, observed during short-term in-laboratory studies, where our study was done at home with no direct observation other than the scale in their bathroom to take a daily weight. This suggests that sleep deprivation effects and these individuals who are only sleeping 6.5 hours a night is a long-lasting effect in terms of controlling energy intake, hunger and satiety. It reverses quickly. You saw it, as Tom pointed out, overnight. One day later, they were sleeping longer, and over that 2-week period, they decreased their energy intake. And this was done in a real life situation. So it should affect the way we live. And yeah.
Did they think to do any cognitive tests of these individuals?
No. There have been some done with sleep deprivation rather than sleep elongation, but there is a decrease in what's called vigilance, your ability to stay with a fixed paper task or monitoring a radar site at an airport if you're a air controller. If you work in the military, if you're doing something, there is a decrease in vigilance. The ability to power through with a hard question is not usually affected until the sleep deprivation goes on for days and days and days. The next step, the one we're working on right now, is to actually test the efficacy of sleep elongation